Okay, welcome everyone. We're in the amphitheater at the Peter Gilligan Center for Research and Learning at the Hospital for Sick Children, and this is our May 2023 issue of Discovery Dialogues, and I'm here with Dr. Ron Worton, who I will introduce to you in a moment. I always have a prop with me, and this is the longest scripted prop I have of any of our guests so far. And that's really representative of Ron's long career and contributions to discovery and administrative science in Canada. We typically don't give a title to the presentations, but Ron actually mentioned a phrase that caught my attention in an email he sent me a few months ago, and that is serendipity in science, unexpected decisions, tackling important research problems. So we'll touch on each of these items as we go through our interview today. And I would also typically script an introduction, but I went to the Canadian Medical Hall of Fame, of which Ron, of course, is a distinguished member. And they had such a nice description, Ron. I'm going to take a second and just quickly run through this. And I quote, described as a role model of one who has achieved success without either aggression or self-promotion, and who brings to every interaction kindness, selflessness, consideration, and empathy. Dr. Worton stands out as both a scholar and a gentleman. Through a novel and groundbreaking approach at a time when disease gene discovery was in its infancy, Dr. Worton and his team identified the dystrophin gene, which is the gene affected in muscular and Duchenne muscular and Becker muscular dystrophy. And then there's a series of key facts, and I'll just touch on a few, and there are many. As I mentioned, Ron's team co-discovered the Duchenne muscular dystrophy gene, and we'll talk about that, enabling definitive diagnosis and prognosis. He led the field in understanding the mutations that are involved in muscular dystrophy. He served as the Associate Director of the Canadian Genetic Disease Network, the head of the Canadian Genome Analysis and Technology Program, and the founding director of Canada's Stem Cell Network. And this is why he's at SickKids. He was a very foundational and important geneticist-in-chief at the Hospital for Sick Children. When I was a graduate student here, he was my boss. His department discovered the genes responsible for Duchenne muscular dystrophy, cystic fibrosis, Tay-Sachs Disease, Wilson's disease, Fanconi Anemia, and at least 20 others in the period that Ron was in lead here. And I think it's recognized that that was probably the most disease genes discovered in any department anywhere in the world. So this was the place to be under Ron's leadership. And then, unfortunately, Ron left SickKids and he went to Ottawa, created a new research institute at the Ottawa General Hospital, and following a hospital merger became CEO and scientific director of the Ottawa Hospital Research Institute and VP of Research. He served on the board and council of over 20 volunteer organizations, including the CIHR Interim Governing Council, and he was the vice president of the Human Genome Organization and a key member in the foundational meetings of the Human Genome Project. And also, lastly, the president of the American Society of Human Genetics. So with that, and there's many other things, Ron, welcome back to SickKids. It's really great to have you here. And I wanted to go back to the start. You're from Manitoba, and tell us a little bit about that and how you ended up with McCullough & Till Stem Cell Laboratory.
That goes back to before this hospital, of course. I did a PhD with Jim Till in the time when stem cells had just been discovered. I went there, I guess, in 1965, and it was a time when they knew that stem cells existed in the bone marrow, mostly in mice, and I had the opportunity to work with Jim on trying to purify stem cells from bone marrow. And it turned out that we weren't able to make them pure by any means, but we were able to distinguish different types of cells, different types of stem cells, show that stem cells were distinct from other kinds of cells that in the bone marrow. So it was an important time for me because it forced me to learn a lot of techniques. And I had come from a physics background. And I think that's why I ended up with Jim Till as my supervisor, because he also came from a physics background in Saskatchewan. And they, in the Department of Medical Biophysics, they tried to place physics students with physics professors or former physicists. So that was a great learning experience, and prior to my coming here.
So let's go back even before that. So I always say that we're in NHL playoff time right now, and all the great hockey players come from Manitoba and Saskatchewan. It seems to be all the great scientists come from there too. What did you learn there? What's your background? What led you into science? What's in your DNA?
I don't think it was a conscious decision at any particular time. It was kind of an evolution. In high school, I didn't know that I was going to be a scientist. I thought probably I was going to be an engineer, and it was pretty much a toss-up whether I enrolled in science or engineering at the University of Manitoba. And I ended up in science in honors physics program. I did 4 years of physics. Halfway through, I knew I wasn't going to be a physicist, and that's when I started evolving into learning something about biology, and ended up in the Department of Medical Biophysics, which is a nice stepping stone for a physicist into biology.
Great. So then you did your PhD here, and then in 1971 you had a big change in your career, I think. What was that about, cytogenetics?
Well, when I left the Ontario Cancer Institute and Jim Till's supervision, I went to Yale to work with a guy who had a very famous name from bacterial genetics, and he was working on fruit flies. And I thought this would be a wonderful opportunity to learn the lessons from bacterial genetics applied to fruit flies. And it turned out to be a bit of a disaster. A lot of the techniques that he was using and proposing to use just were not appropriate for Drosophila. So I finished there, and at that time, Lou Siminovich had been just taken over as the first head of genetics at this hospital. And they hadn't had a genetics department before that. Peggy Thompson had been here and had done a lot of good genetics work, but not under a formal department. So Lou Siminovich took over. And at that time, the cytogenetics lab, which is examining chromosomes in babies, born and unborn, that lab had been run through pathology. And there were some issues there. The lab was way behind in its reporting, needed to be fixed. And so Lou Siminovich called me when I was still at Yale and said, we've got this position open. I think I had written to him in advance and told him I was ready to move back to Canada. And he asked me if I was ready to take over a cytogenetics lab. And I think I said, what's cytogenetics? and so he didn't know either. I had to do some research. But I found a couple of people, including Frank Ruddle, whose name may be on, many of you may know who Frank Ruddle was. And Frank took me into his lab and taught me a lot about chromosomes and mapping of genes. And the clinical cytogeneticist there took me under his wing once a week so that I could do rounds with him and learn about chromosome abnormalities. So, but even then, when I got here and took over the lab, I was pretty green. I had to rely on the technicians in the lab to tell me what I was supposed to do.
Yeah, so I'm the chief of research here, and one of the things I learned from Ron and Lou and others is always recruit the strongest scientists, and the scientists will figure out where they need to move to follow the best and do the best science. So then you did some type of a postdoctoral fellowship with Ruddle, and then you came back here to SickKids, and you could tell us what happened in between, but it was, I guess, 1985, 1986, you started working on muscular dystrophy, and that was a big game changer for you.
Yeah. A little sooner than that, actually, Steve. Probably 1983 was when we started working on muscular dystrophy. Certainly when I came here in 72, so it's quite a long time. And I had been running the cytogenetics lab and prenatal diagnosis lab, and doing research on somatic cell genetics. That was a time when people were struggling to take the principles of molecular genetics from bacteria and move them into human cells. And Lou Siminovich was one of the pioneers in that, and being in his department, it seemed appropriate that I should work in that general area. And that turned out to be critical because the techniques that we developed and that already existed in that era were absolutely fundamental to the muscular dystrophy project, although we didn't know that at the time. How I got into muscular dystrophy was what I meant by serendipity.
Right.
We had a young postdoctoral fellow come to my lab, and she had contacted me from Belgium, where she was a pediatrician and wanted to know if she could do a cytogenetics fellowship in my lab. And I can tell you honestly, the only reason she was looking at my lab was because her husband was coming here for a fellowship in pediatrics and in perinatology. And so she needed somewhere to do her work in the same place where her husband would be located, and I was the fallback. So that's serendipity. But even more interesting is the very first day that she arrived, she sat in my office and told me about a patient, a European patient that she had seen in Belgium that was a young girl with muscular dystrophy. And girls with muscular dystrophy are pretty rare because it's an X-linked disease that affects normally only males. But she also told me that this young girl had a translocation of her chromosomes where short arm of the X chromosome, half the short arm of the X chromosome had been translocated to another chromosome, top of chromosome 21. And she had speculated that the mother might be a carrier and that the translocation might have affected the gene. We knew that the muscular dystrophy gene was on the X chromosome. Maybe the translocation broke the gene into two pieces. And if the mother was a carrier, then the other defective gene would be on the other X, and therefore that would cause the disease. And the piece of information that I knew that she didn't was that in previous women who had X autosome translocations, the other X, the normal X that's not involved in the translocation, is inactive in almost all the cells. And Christine didn't know that, but I did. And when I told her that, we realized that if that was the case, then if the translocation broke through the muscular dystrophy gene and at the same time resulted in inactivation of the other X, then that would cause the disease in a female. And the mother didn't need to be a carrier.
Right.
So Christine's project was took about 2 years to show through techniques that were well known at that time to show that the normal X was inactive in most of the cells. The inactive X is late replicating, so the whole technique was based on showing when the X replicates in the cell cycle. And so she was able to show that the normal non-translocated X was the inactive one. And then we sat on that for, I guess, another 2 years or so, maybe 3, until about 1981 or 1982.
Oh.
More serendipity.
So one of the themes that's come out in these meetings I've had with famous scientists is the importance of trainees, international trainees, access to clinical resources, and over and over again. So that's something we really try to foster here at SickKids. So then, you know, you're a little bit humbled. The techniques were just being developed, and ultimately you found the DNA marker on the X chromosome. So I've been wanting to ask this question for over 30 years. Here goes. So to cut a long story short, for those of you who are watching, the muscular dystrophy gene that Ron's team identified at the time was the biggest gene in the genome. He didn't know it at the time, but it spanned over about 2 million base pairs of DNA. So the first probe that you found, I think it was at this translocation breakpoint, ended up landing in an intron, right? So, and how did that feel when you landed, and kind of like landing on the moon in a crater? You didn't know what to do, where to go, and how did you figure out the next steps?
With your permission, I'm going to go back a step.
Okay, please.
So we knew that the gene was probably on the short arm of the X chromosome, where the translocation had broken the X. But how do you find the DNA from that part of the X chromosome amongst an entire genome? And fortunately, we knew that the translocation in this young woman from Europe, the short arm of the X had been translocated to the top of chromosome 21. And if those of you who know what the chromosomes look like, chromosome 21 is one of 5 different chromosomes that contain a block of genes encoding ribosomal RNA. And we were and then I had another summer student in the lab who was able to show that the breakpoint in her translocation chromosome broke through ribosomal RNA. So we sat on that fact for a little while. And then another piece of serendipity. Do any of you remember the name of Hunt Willard, who was a student not a student, a young faculty member here? When Hunt was job hunting and came to Toronto looking for his first job, I was chair of the search committee. So Hunt and I were walking along University Avenue between here and the Medical Sciences building. And I told him about this translocation and that it broke through the ribosomal RNA genes. And he said, "Oh, I just gave a seminar in Philadelphia, and the group there have just cloned and characterized the ribosomal RNA genes." And so all of a sudden, it became clear that if we could get a ribosomal RNA gene probe, we might be able to use that to fish out the translocation junction from this patient. And so we ended up at Hunt's seminar, and I didn't hear a word of Hunt's seminar because I spent the entire hour sketching out 5 years worth of research to clone the muscular dystrophy gene. Nobody knew that at the seminar.
But that's how it works. And the serendipity is through these interactions, exchanging ideas,
Exactly.
Be in the right place at the right time. But to be in the right place, you have to be out there, exposing yourself. Okay, so tell us about landing in the crater.
All right. So then we use the ribosomal gene probes, but there are about 500 copies of the ribosomal gene in a human cell. And so if you're looking for one translocation junction in a background of 500 copies of the gene, you don't do southern blots anymore, do you? But anybody who remembers a southern blot, you would never find one signal in 500. So we needed to somehow isolate that one chromosome, the translocation chromosome, from all of the other chromosomes in that patient. And here's where my somatic cell genetics background came in, because we were already working with human mouse hybrids and human hamster hybrids that we knew had the characteristic of losing human chromosomes over the course of time. And we also knew that the HPRT gene on the X chromosome was an easily selectable marker. And so we could select hybrid cells. We took the patient cells, fused them with hamster cells, let them lose their human chromosomes, selected cells that retained the X through the HPRT selection system, which we already were using, and we ended up with a hybrid cell that contained only one human chromosome, and that was the X from this woman. And there were only 5 copies of the ribosomal RNA genes on there. So we were looking at a signal of 1 in 5 instead of 1 in 500. And that made it relatively easy to pull out a piece of DNA that had ribosomal RNA gene at one end and a piece of the muscular dystrophy gene at the other end. But I mean, we didn't know for sure that that was part of the muscular dystrophy gene, but we used it as a probe to study boys with the disease to see if it might be deleted or rearranged. And we found 6 boys out of about 100 who had a deletion of that probe, and no normal boys had a deletion of that probe. So we were pretty confident that we were in the muscular dystrophy gene. Now we can come back to your question.
Yeah. That's great. So then what? Because chromosome, you're going to say it was easy. Chromosome walking was not known. There was no reference sequence of the genome for those of you who you couldn't sequence the whole genome.
Well, this was the first gene that had been discovered without previously knowing what the protein was. I mean, the hemoglobin gene was discovered because people knew what the protein was and worked back from the protein sequence to the gene sequence to clone the gene. This was the first one identified without knowing the protein. It turned out that this piece of DNA that we isolated from the translocation junction was in the middle of an intron. I don't even remember which intron
I was going to ask you.
So don't ask me.
It's a big one, a couple hundred kb.
But it turned out that we couldn't find any cDNA that matched that sequence. So Peter Ray joined the lab at that time. He was a postdoc with Lou Siminovich prior to that. And I had just become Geneticist-in-Chief when Lou left. So I now had the power to hire Peter Ray to help me do the rest of the project. And Peter had some experience that I didn't have. So that was good. And with Peter's help, we had to walk the chromosome. We had to find adjacent sequences, purify those, find adjacent sequences to that. And we spent better part of a year, I guess, trying to walk along the chromosome to find a sequence that would be expressed in a cDNA. Mm-hmm. And finally we did, and then we discovered, of course, that then we were able to isolate the cDNA. That was another postdoc in the lab. Even then, we only had about 8 exons of the gene and the corresponding introns. And then Lou Kunkel in Boston at the Children's Hospital had isolated by a technique that I won't go into, another part of the gene, further down the gene. And it turned out that our probes and his probes were all detecting deletions in boys with the disease. So we knew for sure we were in the gene. And I guess it was by 1986. We had the first piece of the gene in 85. We had the full gene in 86, and we had the protein in 87, and some characteristics of the protein by 88. So that was the sort of sequence of events.
And you mentioned Peter Ray's name, and for those from molecular genetics, he was working in Andy Becker's group, and in Toronto there was a phage genetics group was very strong, which played into a lot of the technologies and things.
Yeah, Peter had a lot of molecular technology, and to be honest, I mean, when I got into this, I hardly knew the difference between a gene and a piece of DNA. But that was a lot of people were in that situation at that time.
Even later, no one did.
And so to get me kickstarted, I went to Cold Spring Harbor and did a 3-week course. I was 40 years old. I was the oldest person in the class by 12 years. The next oldest was 28, and the rest of them were kids. And this Cold Spring Harbor course would start at 9 in the morning. We would run until late in the afternoon. We would have dinner. We would have a lecture at 7 o'clock in the evening, and then we'd go back into the lab until around midnight. And then we'd go and play ping pong until 2 a.m. And then we'd get up at 7 the next morning.
Yeah.
And that didn't sit well with a 40-year-old.
So ultimately the gene was identified, and I want to hear a little bit about the prenatal test. I think it was the first, but also just a few minutes on the race to the gene. You mentioned Lou Kunkel and Tony Monaco, and Boston, I think K Davies and Oxford. There were a lot of groups working on this. And how did you deal with the competition? Did you share?
There were 4 groups working on trying to get the muscular dystrophy gene. We all started at about the same time. Luke Kunkel was our main competition. He was in Boston Children's Hospital. And he was working with a specific patient who had a deletion of the gene, a deletion of about 5 or 6 genes. And he was using that patient as his source of material. We were working on the translocation patient. Another person who had the cells from our patient meaning the Belgium patient, was Ed Southern of Southern Blot fame. And Ed was extremely well known in science at that time, way more well known than I was. And so he was using DNA from the same patient. And he also realized, like we did, that there were so many ribosomal RNA genes that he had to purify that protein in some way, had to purify that chromosome in some way. But he didn't have the somatic cell genetics background that we did, so he couldn't do the hybrid technology that eliminated all the other chromosomes. They tried to do it by physical separation, which never worked. He had something like 10 people in his lab working on this project for 2.5, 3 years, and ended up with basically nothing.
Who was the fourth?
And then the 4th one, I knew before you asked me.
That's okay. It'll come back to you.
It'll come back to me.
They didn't come.
No, Kay Davies, of course.
Okay, yeah.
Kay. Kay Davies was doing a more classical mapping approach, like Lepche used for cloning the CF gene. And she had markers that got closer and closer, but she never quite got close enough to the gene. Yeah. And by that time, Luke Kunkel had identified a significant piece of this gene and published in January of 86.
Right.
And we had published in December of 86 another big segment of the gene. So by that time, she was out of the competition.
So that's a good segue into the Department of Genetics, but let's hear just quickly about the prenatal test. I think that was the first in Canada or the world?
So Steve's asking about a prenatal diagnostic test that we did that we never published, and we should have.
Yeah.
I mentioned Peggy Thompson at the beginning. She was the geneticist here in the hospital before we became a genetics department. And her specialty was muscular dystrophy, and she had access to a couple of hundred muscular dystrophy patients that were seen here. She did all the genetic counseling for them. And there was one family that she brought to my attention just within days, or certainly within weeks, of our finding a piece of the gene, long before we had the cDNA or the full sequence. And this is a family that where the mother had a brother with muscular dystrophy, and I believe a brother and maybe an uncle with muscular dystrophy. I don't remember the details, but there were 2 cases in the family, including a brother. And so she was diagnosed as being a carrier through carrier tests at that time. And so she decided, before anybody had the gene, that she would undergo prenatal diagnosis and would terminate the pregnancy of males because they would be at a very high risk, 50% risk of having the disease, but she would carry the pregnancy if it was a female. And she went through that process, I believe, twice. Might have even been 3 times, but at least twice, and had terminated male pregnancies. And then we had a piece of the gene, so we could test her. Now it comes back to me. This goes back a long way. We had studied her DNA, and using our DNA probe from the muscular dystrophy gene, we determined that she was carrying a deletion. So therefore, we would be able to know if that deletion was present in the baby. And by doing the chromosomes, we could tell if it was male or female. So we did the chromosomes, it was male. We did the DNA test, and that piece of DNA was not missing. So we told her that she was going to have a male child, not with muscular dystrophy. And she went ahead and the baby's name was Brandon. I don't know anything. Brandon now would be older than most of you. And so that was a big success story. And I believe it was probably the first prenatal diagnosis anywhere in the world for that disease. And it had to be, because we were the only ones with the probes at that time.
So I make a few comments here before we segue into the Department of Genetics. So you mentioned a lot of names, and just for the audience here, Peggy Thompson and her husband had the Genetics and Medicine book that is utilized around the world for medical genetics training. And then Rod McGinnis and Hunt Willard, who you mentioned, and Bob Nussbaum took over that. And I think, Ron, Ronnie Cohn and myself and Ada Halmash are doing the latest edition. In fact, we just got in our inbox today the print proofs, and many people in the audience helped contribute to that. So we brought that back to SickKids, so you're aware of that. And then also people know, I think under Ron's guidance, Peter Ray started the first molecular genetics DNA lab here, and it's the largest in Canada. And we may come back to that, but let's go after, we'll call it post-muscular dystrophy, into the CF and all the other genes. And that's when I came as a graduate student. And I have my own interpretation, but for you, what was the secret sauce? Why did that department just go to the top? And what was your role? Of course, you were the leader of the department, but what was your interpretation?
There was a meeting held in Asalomar, California, in about 1976, and that's when gene cloning first became feasible. When people for the first time could put a piece of human DNA into a bacterial genome and grow it in the bacteria, and then isolate it from the bacteria after that. It was called recombinant DNA technology, and I guess at that time that was a new term, a whole new idea. Nobody knew if it was going to be safe or not. So the first year or so of recombinant DNA technology was done in underground bunkers with all kinds of precautions that you would now think are totally ridiculous, right? Because there was no risk. But the Asilomar conference was to assess that risk and determine that this was probably a safe technology that could be done in a regular laboratory by anybody. And Lou Siminovich was at that meeting. And when he came back, he held a meeting of all the faculty in the department, not only here at SickKids, but also over at the university. Lou at that time was chair of both the university and the hospital department. And he said to us, this is the way of the future. Cloning of DNA in bacteria to identify human genes is going to be the way of the future. And if you guys want to be part of that future, you have to change what you're doing. And within 2 years, there were 5 of us in the department at that time. Within 2 years, we all phased out what we were doing, in part or in total. And 2 of us went off to Cold Spring Harbor and did that course that I mentioned earlier. And the Lap Chi already had molecular genetic technology from his postdoc, so he didn't have to retrain. Diane Cox did her retraining the year after I did. Manuel Buchwald did it on his own. And we each chose, I was already working on, it was after that I got into the muscular dystrophy, because that was 1976, remember. But within a few years, we all changed our direction. And we were blessed with the fact that some of them, like Diane Cox, was already working on Wilson's disease, right, and had access to all the patients. So cloning the Wilson's disease gene was a bit of a natural for her. Manuel Buchwald had recently shifted his emphasis from cystic fibrosis to Fanconi's anemia, and he was in the process of using different kinds of technologies to determine the number of genes that might be involved, and there are several. And so he was in a good position to clone that gene. So between 86 and 1991, we had cloned a number of genes. Roy Gravel was the Tay-Sachs gene. He was already working on Tay-Sachs, so it was a natural for him. And Steve's point about being probably the #1 in the world was brought home to me when we had a visitor from Oxford, the Oxford professor of genetics, who came to visit me in Toronto to discuss muscular dystrophy and other things. And I walked him around the floor and introduced him to people. And he came back to my office after he'd visited Diane Cox and Manuel Buchwald and Lapchi and so on. And he said, "You're all famous people. I had no idea you were all on the same floor of the same hospital. I knew you were from Toronto, but I had no idea you were all close colleagues." And then he said, "There's no other place in the world that's like this, not even Oxford or Cambridge or whatever." So that was it was interesting to hear that from an outsider who was by that time himself quite famous. John Edwards was his name.
John Edwards. So for those of you who know, that was the 11th floor of the Elm Wing, and the average lab size was probably about 600 to 800 square feet. So we're very spoiled now here in the building. But I actually think that had to play into it. People were packed together, exchanging ideas, interacting on an hourly basis. And do you have any comment on that? I mean, you started your own institute. Yeah. Did you model things after?
Yeah. So Steve's point about we were working in tight quarters with 5 scientists on one floor. All the graduate students on the floor would talk to one another, not just in their own lab, but in the next door labs and the next door to the next door labs.
Yeah.
And it wasn't a very big floor either.
No.
So I think that helped a lot in communication and learning from one another. But I had been through that at the Ontario Cancer Institute in Jim Till's lab.
Right. Yeah.
We were so crowded at the OCI that I never had a bench. I worked in Jim Till's lab for 3 years and I never had a bench. I had a cart and I had all my stuff on a cart and I wheeled it around to wherever I could find space to do my experiments. So in answer to your question, when we started the labs in Ottawa, I deliberately, our labs there were about 1,200 square feet. I deliberately put 2 scientists in every lab to make sure that we wouldn't be spread out too much.
I'm making notes on this. So I didn't mention Ron, of course, is an International Gairdner Award winner, and Lapchi won the Gairdner Award with Jack Reardon, and John Dick, who I think you recruited, who won in the last few years, did his seminal discovery work here at SickKids, and the list goes on. You mentioned many names. It was really a Hall of Fame group and really an amazing time. I'll tell you, I think it was that the quality of people and the mixing and the exchange of ideas. I remember vividly Joanna Romans, who was a postdoc at the time and then an early career scientist, running around at I think it was 4:30 on Thursday afternoon seminars, rounding people up, and people fighting to get in the room for that 4 o'clock or 4:30 seminar. And if you didn't get there earlier enough, you wouldn't get a seat.
Well, it was a pretty small seminar room.
Yeah. Okay. So we got a few more minutes. Is there anything else? I mentioned Peter Ray's name and other trainees. What were your kind of favorite moments from your career? Who were your favorite scientists? Anything else you want to share with us?
Oh boy, that's a big question. I think probably there were two aspects of my career that I will always appreciate the most. One, of course, was the muscular dystrophy gene. That's a hard act to follow, and I knew that I wasn't going to do another thing like that. In fact, when I left here, I was recruited to Ottawa. I began to look around because I was in the 11th year of my 10-year term as head of genetics. And I knew that carrying on in that role was not the right thing for me to do, nor the right thing for the hospital to do. And as luck would have it, Ottawa was looking for somebody to create a new research institute patterned after SickKids. And so when I applied for that, I didn't apply. They approached me and I went and interviewed. And when I came back and told my colleagues here that I was going to move to Ottawa, the reaction from most of them, who were all on University Avenue, said, Ottawa? Why would you go to Ottawa? This is Mecca here, right? And it turned out that it was probably the next best thing I could have done in my life, because I went from being a medium-sized fish in a large pond to being a big fish in a small pond, right? And I was able to create a research institute there that during my time went from annual funding of about 5 or 6 million up to 85 million. And we recruited about 100 scientists, patterned after SickKids, all the same characteristics, and tried to organize it in the same way that SickKids was organized. One of the things that I had always believed in for hospital-based scientists is no tenure. And SickKids, we didn't have tenure. Is that still true here now?
Yeah.
And when I got to Ottawa and said, we're going to recruit people with no tenure, even the university was surprised and didn't think that was right. But it turned out to be absolutely right, because it's a defining characteristic of people whose livelihood depends on doing research.
Yeah. So we're going to wrap up, but we have an audience here, and I'm going to invite them to ask questions, but that'll be at the very end. I do have one more question. We have a photograph it's in a special display case just up here. We looked at it on the way down, and it's a picture of Ron attending really one of the very first, and I think the most important foundational meetings of the Human Genome Organization Committee. And Ron was on that committee representing Canada. And that was very important in my own career. I remember when you came back from that meeting and you gave an update in the old auditorium in the hospital. And I sat in the 13th row and I listened, and it really captured my attention. But in that picture, of course, James Watson, Wally Gilbert, there's 6 Nobel Prize winners, yourself. Charles Schreiber was on the committee, but he was not at the meeting, and Charles just passed away last week or so. What was it like there, and just setting the stage for the next, many of us in the audience are genome people. So you set the stage for the next generation of science. What was the feeling like?
Yeah, so I was sort of the little kid on the block at that meeting in a way. We were still working on muscular dystrophy at that time. And I believe it was Victor McCusick who actually called the meeting. Victor was not a molecular biologist, but an extremely well-known geneticist who tracked genetics all over the world and had probably the world's best databases of genetics. And Jim Watson was at the meeting, and many others of great fame. And the value of that meeting was that up until that point, there were 2 groups of scientists that didn't talk to one another. There were what were called the gene mappers in the Frank Ruddle mold, who were busy making somatic cell hybrids and segregating chromosomes and mapping genes to chromosomes. And they would interact to some extent with the human geneticists who were mapping genes through family studies. And then there were the molecular geneticists who were cloning pieces of DNA, but those 2 groups never talked to one another. And so in terms of genome, there was one set of publications related to mapping of genes and another set related to cloning of genes, totally different groups of people.
Right.
And that meeting in...
we think it was in France.
That meeting brought together.
Montpellier, right?
Yeah.
I think it was Montpellier. Is that France?
I'm not sure. Anyway, it brought together the two groups of people who talked for the first time and decided that they would create something called HUGO, the Human Genome Organization. So we spent a couple of days defining what this organization would look like, what the membership would look like, how you would become a member, what its goals would be, what the annual meeting would look like, and all of that. And then coming out of that, to my surprise, I was became part of the founding council, I think it was called.
Right.
And then a couple of years later, I was vice president for the Americas, which was something new for me. I had to worry about not what was going on in just North America, but also South America. And it wasn't much, but I had to pay attention to that. Interesting time.
Yeah. So just finally, I wanted to thank you so much, Ron, and thank you on behalf of SickKids and all of Canada for everything you've done for science over the years. And I'll just leave you with one last thought. I remember at some point in my career, Ron told me that, at some point you have to just end it. And he said he was going to retire at 65. I think he went a little bit longer than that. And he said he owed it to your wife and your family to go back and spend time with them.
I retired at the end of the month in which I turned 65.
Wow. I don't know how you could have accomplished so much in that time. And I think you started early, but certainly it's because of your hard work and intelligence. And thank you so much for the interview today.
I'll just add one more little parting thought. Part of the reason for retiring at 65 was, I wanted to do something different. I wanted to do something totally different. But I had also seen two instances of people hanging onto their jobs too long and getting in the way. And I didn't want to do that. And so this was an excuse and an opportunity.
And you bought a boat and had a lot of fun.
We had the boat before that. Yes, we had a lot of fun on our boat.
Okay. Thank you again, Ron. And thank you for attending. And if anybody has any questions, stick around. And we're actually meeting with Ron, having lunch. And we've invited people from what we call the Genome Clinic to present how we do cytogenetics in 2023, which is just a sequence of genomes.
Yeah.
And we'll give you an update on what's happened in the last few decades.
Good.
Thank you very much. Great. Thank you. If anybody has any questions, come on down. We've got a few minutes. Introduce yourself to Ron. That was great. I don't know what is going on up here, some other meeting. Could you hear